One. Yay.
You're so mean.
Alright. What?
You're
No.
bully.
Okay. I'm not putting it online because no one pays attention.
What? What are you talking about? It's going to be online. It's already online. Is it? This is online. Oh, yeah.
Okay.
Congratulations.
Anyways, DNA. That's our first topic. Today is Unit 5, which is all about information. And so it's passing information going from DNA to RNA, or going from DNA replication, or going from all of that. So development. And so we're first talking about DNA. Deoxyribonucleic acid. It's going to be, what do you know? A nucleic acid. That's going to be the type of polymer that it is, or the type of macromolecule. Its monomer, or its building block, is going to be a nucleotide. And all these nucleotides have the same basic structure. All of them have a phosphate. All of them have a pentose sugar. And all of them have a nitrogenous base attached to them. Which nitrogenous base they have could be different. There's 4 different types. We can have A, we can have C, we can have T, and we can have G. It's out. You want to open the door? Thank you. Okay, so there's 4 different things that can be there. For the phosphate, it's all the same. The sugar is going to depend on whether or not I'm looking at deoxyribonucleic acid or I'm looking at ribonucleic acid. If it's RNA, it's got a ribose sugar. If it's DNA, it's got a deoxyribose sugar. Following along, niños? Ooh, that was sweet. No, it's just the old peach or grapes. And then, if I look at it, how DNA is structured, do you remember what the structure is called? It's called a double helix. So it looks like this. It looks like a coiled up, twisted ladder. Okay? And on one side, it's going to go from 5' to 3'. On the other side, it's going to go from 3' to 5'. So because I have that, one side's going from 5 to 3 and the other side's going 3 to 5, we've got a term for this. These strands are parallel, but they run in opposite directions. And so what that's called is anti-parallel. Kind of like the freeways. Okay. And if I look at it on a molecular level, how it actually looks is I would have a P, S, P, S, P, S. It's going to look like this. So my sugars and phosphates are going to make up the sides of the ladder. So S, 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 P, P, P. Alright, so those are my sides of my ladder. On the insides of my ladder, I have my nitrogenous bases, or my A's, C's, T's, and G's. So this is what my DNA looks like right here. So my rungs of my ladder are my nucleotides. My sugars and phosphates are going to be my sides of my ladder. Okay. And remember, as I'm doing this, the percent of A's is going to equal the percent of T's, and the percent of C's is going to equal the percent of G's. So if I were looking at the percent of A's, and let's say the percent of A's is 14%, what would the percent of T's be? 14. What do you know? 14%. Okay? Those collectively equal 28%. Meaning 100 minus 28 is going to be 72. Meaning C plus G has to equal 72. So that, and then I would divide that because they equal each other, meaning C is going to be 36% and G is going to be 36%. Because collectively, all of this now adds up together. 14 plus 14 plus 36 plus 36 equals 100. 100. Not 100, 100. Thug life. Alright. You following along? Any preguntas? Sorry. I just see lots of heads turning, so I'm waiting a second. Now let's start talking about DNA replication. if I look at it, so we have a specific model that we have for how DNA gets replicated. It's called the semi-conservative model. So it's a semi-conservative model, meaning if I were to take the parent strand of DNA, let's say it looks like this, and I said blue equals old, and I replicated it. Okay, what it's going to do is each half of that parent strand is going to serve as a template. So it's going to get uncoiled. Okay, helicase is going to come along and unzip it, and each half is now going to serve as a template for a new strand. And then I'm going to have new DNA added that's complementary to it. So half of that strand is old, half of that strand is new. Okay? That's why it's called the semi-conservative method. Because half of it's old, half of it's new. True. Oh, I didn't know you named that soup after her mom. No, Chunky. I don't even know your mother. I do bet she's a nice lady.
She doesn't like you though.
I don't blame.
Like she actually doesn't like me.
It's okay. There are lots of people who don't. Okay. So what I have here is, if I take my DNA and I look at it now on a closer level, how this gets replicated, we form lots of these things. They're called origins of, origins of replication, where replication starts at. And it's going to go in both directions. I can have something right here and I can have something right here. What it's going to do is, these are going to be known as helicase. This helicase is going along and it's unzipping the DNA. And as it unzips it, it forms something. It forms what's called a replication. Yep. A replication fork. Because it's where there's like a split. It starts as one and it splits, kind of like a fork in the road. So that's what happens at this. And I can have multiple of these origins of replication along the DNA. That way DNA gets replicated faster. Helicase unzips it, forms a replication fork, and we keep on keeping on. What I'm going to do now is I'm going to take this and I'm going to zoom in a lot more. Okay? So if I were to zoom in, did you guys still need that? I'll give a couple more seconds.
Thank you.
You're welcome. Those are pretty dope notes. I'm kind of impressed. All right. Replication fork. All right, we're moving on. Now I'm looking at it. Let's say here's my DNA again. And I'm zoomed in now to that replication fork. So remember, I had that enzyme helicase. I've got this area right here called a replication fork. Sorry. Do you want me to go back and fix it? Okay, I've got a replication fork. If I'm looking on these strands, on this strand right here, it's going to be 5 prime. This strand's 3 prime. So that means this is 3 prime and this is 5 prime. On my original parent strands. Okay? What happens when I very, very first start this? Something's got to get put down way up here. It's called an RNA primer. Okay? And the enzyme that puts that down is called primase. It's going to lay that primer down. It's an RNA primer that gets put down. And now what's going to happen is I'm going to have another enzyme come along, and it's going to start adding complementary nucleotides. It's going to add complementary nucleotide to the growing 3' end. So as helicase unzips, all right, it's going to now expose this 3' end, so it can add more nucleotides and add more nucleotides and add more nucleotides. This strand right here is now called the leading strand. Okay, because 5', 3', I can only add nucleotides to the 3' end. So as helicase unzips it, it exposes 3'. Unzips more, exposes 3'. Unzips it, exposes 3'. The other strand, not so much. Because on this side I've got 3', on this side I've got 5'. So it can only add in the opposite way. It can only add going to the right, where on ours it was adding going to the left. So what it has to do is it's going to unzip a little, lay a primer, and then come along and it's going to add nucleotides. Oh, I got to go back. Oh, man. Lay a primer, add nucleotides. Okay? Lay a primer, add nucleotides. So primer, nucleotide. Primer, nucleotide. Oh. N for no. Okay. And so what it did now is now it created these little fragments. It's going to then, we have a name for these fragments. Do you remember what they're called?
Uncle Rocky's fragment.
Yes. Okazaki fragments. Because I've got the 3' end isn't exposed, as I unzip it more and more, all I do is expose a 5' end. So it's going to unzip it a little bit and then go backwards. Unzip it a little bit and then go backwards. Okay? Eventually, ligase is going to link all these fragments. We're going to replace those primers with DNA, and then ligase is going to come along and it's going to link all these fragments together because ligase links. All right? And now we're left with 2 daughter strands that are identical to each other. All right? Any questions? The enzyme that came along, remember, and added these nucleotides, since I didn't really write it. Let's say this is the enzyme that adds the complementary DNA nucleotides, is going to be DNA polymerase. Oh, that was terrible. DNA polymerase 3. It's going to add the complementary nucleotides to the growing strand. Okay? Any questions on DNA replication? And so there's one last thing that we got to talk about for DNA replication, or not DNA replication, but for DNA in general. I should have talked about it earlier. Transformation. Okay. Anyone remember what transformation is? We did it when we did the Piglo lab. It's where you insert a gene into a new cell. So for instance, for us, we took a gene for Piglo. All right, we took a Piglo plasmid. And we inserted that plasmid into that. So let's say I've got a sequence right here of DNA, and this is my gene of interest that I want to put in the red one. What it would do is it would take that and it would insert that gene right there. And so now I've got a sequence of DNA or a cell that has a new gene in it. So now it's got new traits. That's what transformation is. That's how we were able to make E. coli bacteria resistant to antibiotics and to glow underneath UV light. It's fascinating, I know. Hold on to your britches, kids. Now let's talk about how we make that there protein. So if I want to make a protein, I've got to undergo transcription first. Transcription's going from what to what? DNA to mRNA. All right. And in eukaryotes, you remember where it happens at? Nucleus. In prokaryotes, cytoplasm. Or cytosol. Okay, and there's 3 big steps for this whole thing. So it's basically, if I were to take DNA and I had like A, C, T, G, C, G, A. First off, how do I know that's DNA? Because that's a T. That tells you right there it's a DNA. And if I were to make the complementary mRNA strand, it would be U, G, A, C, G, C, U. So that's the complementary mRNA strand. Okay? Because now I have U's, I don't have T. And so now let's talk about the 3 steps. Step 1, initiation. What I have in initiation, transcription factors bind somewhere. All right, it's called a promoter sequence. So, promoter. Gianna, you remember what that most common promoter is in eukaryotes? What kind of box? Tata box. Yep. So it's a Tata box. And when it does this, it forms something called a transcription initiation complex. Okay, transcription initiation complex. And now for step 2, elongation. Okay, during elongation, RNA polymerase adds RNA nucleotides to the growing RNA, or to the mRNA strand. You know what the last step is? Termination. The transcript is released. And now we've got to process it. Gots to do some stuff to it. Let you guys just sit there and look at it and take all that in. Okay, so now we go to mRNA processing. I need a thinner black one. mRNA processing. This happens in the nucleus, so does that happen in prokaryotes or eukaryotes or both? It only happens in eukaryotes because prokaryotes don't have a nucleus for this to take place in. So this is only eukaryotes. Okay. And so there's a couple things that are going to happen here. The big one first, we're going to add a poly A tail to the 3' end. So it's going to have like 50 to 250 of these things on the end of this thing. The second one, 5', we're going to add a GTP cap. Okay. So 5' is going to be a GTP cap. And then finally the third one, RNA splicing. Meaning introns are removed, yes, introns removed. How you'll see this a lot of times is you'll see it look like this. You'll see it look like this, and you'll see this will be labeled I1, E1, I2, E2, I3, E3. Okay. Remember, which part of these get pieced together so they can leave the nucleus? The introns or the exons? Okay, the exons. So, if I were to look at this, it would end up looking like this. I would get rid of all those introns, and I would piece together E1, E2, E3, and E4. I would piece all of those together. All the ones I have are over there on one of the shelves. You'll see them. I don't know how many there are. Okay? So it pieces all those together. This is before, after. You can check. There might be some on the whiteboard lit too. I don't know how many there are. Oh, there look like there's 10. Cut. Are you ready? Well, that's right there. So now that this has happened, I'm ready to process this. I'm ready to go to translation. And that happens at the ribosome. The ribohomies. Okay. There's 3 parts for this. Step 1, initiation. Okay. It's going to have a reading frame. And you're going to have a start codon. And remember, a codon equals 3 nucleotides. Okay, codons are going to be read in chunks of 3, or nucleotides are going to be read in chunks of 3 called codons. One of those is going to code for methionine, and it's going to be the start codon. That's initiation. When that happens, the tRNA is going to bring the first tRNA. First amino acid. I'm going to abbreviate amino acid AA. So tRNA is going to bring the first amino acid. And how that tRNA looks, it's going to be going to look like this, and it's going to have that attached to it right there. This is the AA. Okay, that's the amino acid. It's going to have letters right here, so it might have C, G, C. It's going to read this it's going to read the mRNA. It's going to want mRNA that's complementary to it, so it'd be G, C, G. They match up with each other. On this little collection right here, is known as a codon on the mRNA. Okay, and this one right here is known as an anticodon on the tRNA. Okay, so that's how it's going to know which amino acid to bring. It's going to pair up with that mRNA. So the mRNA is going to have a codon, the tRNA has an anticodon, and it's going to be very specific. It's going to say, hey, bring this amino acid. So that way it pairs up with it. Because remember, C pairs with G, G pairs with C, C pairs with G, A pairs with U. So that's how it's going to know which one it needs to bring in. So you need to be familiar with how to read your codon chart tomorrow also. So that little chart where you go like A tells you it's in this row, C tells you it's in this column, that stuff. You need to be familiar with that. Okay. Step 2 is elongation. Okay. In elongation, we've got on the ribosome there's 3 sites. There's the A site, there's the P site, and there's the E site. Okay. That A site is where the tRNA, so new tRNA approaches with an AA, with the AA that needs to be brought in. The P site is where I'm going to have my polypeptide. So I've got my growing polypeptide chain sitting in the P site. The A site's going to bring in a new tRNA molecule with an amino acid on it. That polypeptide is going to break off and join onto that new one. All right? So the P site's where I kept my growing polypeptide chain. And now what's going to happen is everything's going to shift over 1. So originally, I had my growing polypeptide in the P site. The new tRNA approached at the A site. Now that big chain breaks off and adds itself onto the new amino acid, and now everything's going to shift over 1. And so in that E site now, the empty tRNA exits. Okay? And so it's just going to keep doing this over and over and over again, and growing and growing and growing, until I reach step 3, which is called termination. Which is stop codon is reached. Polypeptide released. Okay. Oh, I'm sorry. Yes. That good? We have a whole nother page. We've got to learn about mutations in cell cycle. Okay. So now that I've made this, we've got to talk about how can I mess it up? Because there's ways to. Mutations. Okay? There's one of the big categories of mutations is called point, which are also known as substitution. Okay, substitution mutations. And there's 3 big types here. The first one is called a missense. In a missense mutation, anyone remember what happens there? That's a silent. So in a missense, that's a nonsense. There's one more.
Changes the amino acid.
It gives the wrong amino acid. So it codes for the wrong amino acid. Okay. In a silent, okay, nothing happens. In a nonsense, we get an early stop. Okay, we get an early stop codon. But then we can also have frame shift. Frame shifts are insertion and deletion. We insert a nucleotide or we delete a nucleotide. I feel like those are pretty self-explanatory based on their names. Missense. A missense can be bad, it can be neutral, so I'm just going to put like a I don't really know face. Silent is a good one. Because yes, I have this mutation, but nothing happened. Everything's still peachy keen. A nonsense mutation is bad. Not going to have a good time. Okay, nonsense mutation, we have an early stop codon, so it's bad.
Rip.
Rest in peace, homie. Frame shifts are all bad. If I were to delete 2 nucleotides, dunzo. Everything changes. Throw in a nucleotide, everything changes. Delete a nucleotide, everything changes. Bad news bears. Cut. That's mutations. No. Because we're done with protein synthesis. Now we have cell cycle. You have the flu? Okay. Cell cycle. There's 2 big types of division. The first one, mitosis. In mitosis, it's dividing somatic cells. Meaning it's going to be cells that are diploid. Or 2N. Meaning they have 2 copies of every single chromosome. In mitosis, it only divides once. Resulting in how many daughter cells? 2 daughter cells. And those daughter cells are identical. Because mitosis is used for growth, repair, replacement, anything like that. So we want those cells to be identical. Compared to the other type of cell division. You remember what that other one is? Meiosis. Okay. So meiosis. And so meiosis happens in gametes. Or sperm and egg. Okay? And those gametes are haploid. Meaning they've only got 1 copy of each chromosome, or so they're in. Okay? All of our somatic cells have 2 copies, 1 from mom, 1 from dad, for a total of 46. In our gametes, we've only got 1 copy for a total of 43 chromosomes. Because those gametes are eventually going to combine with another gamete, so that's why we don't want 2 copies in there. In this, meiosis undergoes 2 divisions, so it divides twice. How many daughter cells do we end up with? 4. Divides once, you got 2. Divides twice, you got 4. Divides 3 times, you got 8. Divides 4 times, you got 16. So that number just doubles. Are they identical?
No.
They're different. They have half the DNA. Okay? And what meiosis does, meiosis increases variation. So it increases genetic variation, and there's 2 ways it does that. Way number 1 is called crossing over. Crossing over is when you've got 2 gametes, let's say right here, or 2 homologous chromosomes. I've got that one, and I've got this one. Remember, they're going to pair up. But part of them is going to swap. Okay? So they take a tiny bit of their DNA or part of that chromosome and it swaps. So crossing over is going to be this small section right here, because if you look, they swapped. It'd be like if I crossed with someone else, let's say Demetrio, and Demetrio and I swapped hands. Okay? I've still got the rest of my body and everything else, but now I've got his hand on my body and he's got my hand on his body. That sounds bad. Okay? The next one is going to be... Ooh. Oh. Independent assortment. What that's going to be is, let's say I take a cell. When I have this cell, these chromosomes are going to line up. Okay? And they're going to pair up in homologous pairs. Whether they line up on the left or line up on the right for chromosome 1 has no effect whatsoever on chromosome 2. So let's say I've got chromosome 1 goes left, right, right, left, left, right. Has no effect whatsoever on it. Alright? So it's completely random which side they line up on. But it makes all the difference because when that cell eventually divides, if it lined up on the left, it goes to the left. If it lined up on the right, it goes to the right. So what happens in this cell is completely random. Alright? And so that's going to add to variation. Okay? That's two ways we get variation. That's why you don't look the same as your brother and sister. Maddie, I'm sorry about that. Your brother's a good-looking guy. Okay. So the cell cycle. I've got a couple stages. So if I look at all of the cell cycle, my very, 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 very first stage is going to be called G1 or gap 1. And what happens there is growth. Okay? After G1, something happens before the S phase. A checkpoint. Okay? It's going to make sure everything's ready to go before replication. If it's not, it goes to another stage. It goes to what's called G0 or G0, which is no division. Okay? If everything's good to go, now it's ready to go to the S stage for synthesis. Okay, so during that stage, DNA is replicated. Okay, DNA gets replicated. There's no checkpoint after this, but now it's going to go to the G2 stage, which is gap 2, which, more growth. And then I'm going to have another checkpoint. It's going to make sure everything was good. If it is, it's ready to go for division. So all of that stage collectively, all of this is known as something. Do you remember what it's known as? Interphase. Known as interphase. Let's say I'm looking at DNA numbers. Let's say we did what's called an assay. It's where you can actually measure out and weigh out how much DNA there is in each thing, and it's measured in picograms. Let's say for G1 stage, I've got 10 picograms. Okay? Or as third period said, 10 picodegaios. I've got 10 picograms. During the S stage, how much DNA would I have? Would it still stay at 10? No, it would double. And during the S phase, I would go from 10 to 20 picograms because that's where my DNA gets duplicated. During G2, I would stay at 20 picograms. After G2, what's the next stage? After G2, it's mitosis. Okay? But something's got to signal it to happen. There's a certain chemical that starts increasing in levels. okay, that says, hey, it's time to divide. This mitosis promoting factor, or MPF. MPF goes up, and now it's ready to go into the M phase, or mitosis. During mitosis, the nucleus divides. Okay? And then we have another checkpoint. And then after mitosis, something else, there's one last thing that has to divide. The cytoplasm. So cytoplasm divides. It can either be through cleavage furrow, or cell plate. Okay, now back to these numbers. During mitosis, what do you think happens to my DNA? Does it stay the same? No. It goes from 20 down to 10. And same with right here, it stays at 10 picograms. So if I look at, right there. So if I look at my DNA levels, my DNA is going to go G1, S, G2, M, G1, S, G2, M, okay? So, okay, so that's what's going on there. So we have all these checkpoints and everything to make sure these cells are dividing when they need to divide. Sometimes stuff can get messed up. And these cells don't do these checkpoints right, or they don't stop dividing when they're supposed to stop dividing. And it forms something. It forms a tumor. All right? We can have benign tumors or we can have malignant tumors. And what happens is if it's a malignant tumor, it forms something called cancer. Okay? And what happens with cancer is it's caused by mutations. And there's normally something called a density-dependent inhibition. Meaning, if I've got two cells, and these two are right here, all right? I've got these two things. They touch each other, so it tells it, stop. Okay? Once they touch another one, they have surface receptor proteins, and it says, hey, stop. If I've got cancer, there's a mutation on that. It doesn't know when to stop. All right? And so it just keeps doing it and keeps doing it and keeps doing it. All right? And these checkpoints aren't doing anything to stop that. That's what cancer is. It's unregulated cell growth. Okay? And our last one that we've got to talk about is develop, and the category is development.
Do you think I could fit it right here?
Yeah, all it is the two types of stem cells, adult and embryonic stem cells, and then what is cloning. Okay, so for development, we've got two types of stem cells. We can have embryonic, or we can have adult stem cells. You remember the difference between the two? Embryonic can give rise to any type of cell completely. And there was a term for that. It was totipotent. Meaning it can give rise to any type of cell. Where adult stem cells cannot. They can give rise to a lot of them, but not all of them. So they're called pluripotent. Adult stem cells are called pluripotent. Okay. Last one, cloning. Or what's known as nuclear transplantation. What it does is I'm going to start with a cell right here that has 46 chromosomes. If I were doing this with a human, a somatic cell. Okay? And I'm going to take another egg cell. All right? And normally an egg would have 23 chromosomes. I'm going to take that and I'm going to get rid of that nucleus. So now I've gotten rid of this. I've denucleated it. And what I'm going to do is I'm going to take this nucleus and transplant it into this, giving me an egg cell with this nucleus that has 46 chromosomes in it now. Now I've got an egg with a full set of DNA in there. And I just have to take this egg, insert it into a surrogate, or a mom essentially, and now it's going to give rise to a clone. That clone is going to be identical to this the organism that gave this somatic cell. So, if I took an egg from Izzy and a skin cell from Demetrio, got rid of the nucleus in your egg, put the nucleus from your skin cell into her egg, now I would have a genetically identical copy of you. Even though it's her egg, it's got all 46 of his chromosomes in there. Normally we would get 23 from mom, 23 from dad, but this time we got all 46 from dad. So there's no variation whatsoever because there's no independent assortment, there's no crossing over, there's no anything. All I've got is my own little Demetrio. It's a good life. Okay, any questions?